Right now, as you lie there listening, you are not alone inside your own body. You are inhabited. Not in a frightening way. Not in a dramatic way. But in a biological, measurable, astonishing way. Trillions of microscopic organisms are living on you and inside you at this very moment. They are breathing, dividing, signaling, digesting, and communicating. They outnumber your human cells in certain regions of your body. They influence your immune system, your metabolism, your mood, and possibly even your thoughts. You are not a single organism. You are an ecosystem. And tonight, we are going to gently explore the hidden world within you. Before we begin, leave a quick comment and tell me where you're listening from. Somewhere across the planet, under another sky, someone else is learning about the invisible life inside them too. Now take a slow breath and imagine shrinking down, smaller than a grain of sand, smaller than a single skin cell, until you are entering the microscopic world of the human body. Inside your gut alone, there are trillions of bacteria. The colon is one of the most densely populated microbial environments on Earth. In a single gram of intestinal content, there can be more microbial cells than there are people on the planet. These microbes form what scientists call the gut microbiome, the complex community of bacteria, archaea, viruses, and fungi that coexist in delicate balance. They are not invaders. Most of them are partners. When you eat food, your body begins digestion in the mouth and stomach. But many complex carbohydrates and fibers cannot be fully broken down by your own enzymes. Instead, they pass into the large intestine, where microbes ferment them. In this process, they produce short-chain fatty acids such as butyrate, acetate, and propionate. These molecules nourish the cells lining your colon. They regulate inflammation. They influence metabolic health. Without your microbiome, digestion would be incomplete. But their influence goes further. Your immune system is constantly interacting with microbial signals. In early life, exposure to microbes helps train immune cells to distinguish between harmless substances and dangerous pathogens. A balanced microbiome supports immune regulation. When microbial diversity declines, inflammatory disorders become more common. Inside your gut, immune cells and microbial metabolites exchange chemical messages constantly. It is a conversation. And you are both the host and the habitat. The microbes in your body also produce neurotransmitter precursors. Certain gut bacteria are involved in synthesizing molecules related to serotonin, dopamine, and gamma-aminobutyric acid. While the brain produces its own neurotransmitters, the gut microbiome contributes to signaling along what scientists call the gut-brain axis. The vagus nerve, a long cranial nerve extending from the brainstem to the abdomen, acts as a communication highway between gut and brain. Signals generated in the gut can influence mood, stress response, and behavior. Your emotions are not caused by microbes. But microbes may influence the chemical environment in which emotions unfold. The relationship is subtle, bidirectional, complex, and still being studied. Even your skin hosts microbial communities. The surface of your skin contains billions of bacteria adapted to specific microenvironments. Oily regions host different species than dry regions. your microbiome shifts depending on climate, hygiene, age, and environment. These microbes help defend against pathogens by occupying ecological niches and producing antimicrobial substances. They form a protective layer that discourages harmful organisms from taking hold. You are never sterile, and that is healthy. Inside your mouth, microbial communities form biofilms along teeth and gums. In your lungs, low-density microbial populations exist in ways that were once thought impossible. In your reproductive tract, distinct microbial ecosystems support tissue health. Each region of your body is its own habitat, and these habitats are dynamic. When you are born, your microbiome begins to assemble. Mode of delivery, vaginal birth or cesarean section, influences initial colonization. Breastfeeding introduces beneficial bacteria and nutrients that shape microbial growth. Early exposure to soil, animals, and other humans increases diversity. Over time, your microbiome stabilizes into a relatively unique signature, almost like a fingerprint. No two individuals have identical microbial compositions. And yet, within that uniqueness, there is balance. When balance shifts too far, a condition known as dysbiosis, health can be affected. Antibiotics, while life-saving, can temporarily disrupt microbial communities. Diet strongly influences composition. Fiber-rich diets support diverse microbial populations. Highly processed diets may reduce diversity. The ecosystem inside you responds to what you consume, and responds quickly. Within days of changing diet, microbial populations can shift measurably. It is a living, adapting system. The number of microbial genes inside your body vastly exceeds the number of human genes in your genome. While the human genome contains roughly 20,000 protein-coding genes, the collective genomes of your microbiome contain millions. This expanded genetic capacity allows microbes to perform biochemical tasks your own cells cannot. You are genetically collaborative. A human-microbe partnership. And this partnership has existed for hundreds of millions of years. Microbes were present on Earth long before animals. When multicellular organisms evolved, microbes evolved alongside them. Symbiosis, close biological interaction between species, shaped evolution itself. Mitochondria, the energy-producing structures inside your cells, originated from ancient bacteria that formed symbiotic relationships with early eukaryotic cells billions of years ago. In a very real sense, you carry ancient bacteria inside every cell. The boundary between you and microbe is not absolute. It is layered, fluid, interdependent. If you imagine your body now not as a single entity, but as a vast landscape filled with microbial communities, valleys of bacteria in your gut, forests of microbes on your skin, delicate ecosystems in your mouth, something changes in how it feels. You are not isolated. You are inhabited. And that habitation is not hostile. It is cooperative. Trillions of microscopic lives working continuously to help digest food, regulate immunity, prevent infection, and influence biochemical balance. They do not think. They do not plan. But collectively, they shape your health in ways that science is still uncovering. And as you lie here now, breathing slowly, those microbes continue their work. fermenting fiber, producing vitamins, competing with pathogens, sending chemical signals through your tissues. Quiet, invisible, essential. You are not a single organism drifting alone in the night. You are an ecosystem. And that ecosystem is calm, balanced, alive. Right now, as you lie still and breathe gently, there are trillions of lives moving inside you. They do not sleep when you sleep. They do not wake when you wake. They simply continue, dividing, fermenting, signaling, adapting, part of a system so constant that you rarely notice it at all. You are not just one organism drifting quietly in the dark. You are a living landscape. If we could shrink down to a scale smaller than dust, smaller than a single cell, and drift into the warm corridors of your body, we would not find emptiness. We would find motion. A dense, crowded, humming world, especially within the long winding passage of your gut. There, in the soft darkness of your colon, microbes gather in numbers that are almost impossible to imagine. In a single gram of intestinal content, there can be more microbial cells than there are human beings on Earth. And yet this is not chaos. It is organized. These microbes do not float randomly through your body. They settle into niches. They form communities. They build structures. Some cling to the protective mucus lining of your intestinal wall, others drift through partially digested food, and still others cluster together in layered colonies that resemble microscopic cities. When you eat, the story continues. Your own enzymes break down much of what you consume, but certain fibers and complex carbohydrates pass untouched into the deeper parts of your intestine. There, the microbes take over. They ferment these fibers slowly, releasing molecules that your own body could never have produced alone. Butyrate, acetate, propionate. These small compounds drift outward, nourishing the cells lining your gut and helping maintain the integrity of the barrier that separates your inner world from everything passing through it. The border is important. Your gut is technically an open tunnel to the outside world, and yet you do not dissolve into everything you swallow. A thin layer of cells, tightly joined together, forms a careful boundary. Just beyond that boundary, immune cells stand watch. Not tense, not alarmed, but attentive. They are constantly sampling signals from the microbes nearby, learning which patterns are harmless and which require action. The conversation between your immune system and your microbiome never stops. It begins early in life, when the first microbes settle into place and your immune cells learn the difference between friend and threat. It continues through childhood, shaped by food, soil, contact with other people, the air you breathe. It adjusts quietly every time your environment shifts. The goal is not sterility. The goal is balance. Inside your gut, chemical messages move steadily along the walls and into the bloodstream. Some microbes produce compounds that influence how your body regulates inflammation. Others modify bile acids, subtly shifting how fats are processed. Still others participate in synthesizing precursors to neurotransmitters, molecules that help regulate communication within your nervous system. There is a long nerve, the vagus nerve, that stretches like a quiet highway between your brain and your abdomen. Signals travel in both directions along this path. The brain influences digestion, The gut sends chemical updates back upward. It is not control. It is dialogue. If your days are irregular, if sleep is shortened, if meals come at unusual hours, the rhythms inside your gut shift as well. Microbial populations fluctuate depending on when nutrients arrive. The tide inside you rises and falls according to patterns of light and dark, feeding and fasting. Even your microbes participate in circadian rhythms, responding to the timing of your life in ways that are still being mapped. The surface of your skin tells another story. Your skin is not a sterile shell. It is a mosaic of climates, dry plains on your forearm, humid valleys in the bends of your elbows, oil-rich regions across your forehead. Different microbes prefer different conditions, and so they settle accordingly. They compete quietly with invaders, occupying space and producing antimicrobial compounds that discourage more dangerous organisms from taking hold. Even your mouth hosts a structured society. Along your teeth and gums, bacteria form biofilms, organized layers embedded in protective matrices. When balance is maintained, this biofilm coexists with you. When sugar intake is frequent and conditions shift, the chemistry changes and enamel can slowly erode. Again, not because microbes are villains, but because ecosystems respond to environment. The same is true deeper inside. Viruses that infect bacteria, bacteriophages, drift through your microbial communities, shaping which bacterial populations expand and which contract. Fungi contribute their own subtle influence. Archaea, ancient microbial lineages, process fermentation byproducts in ways that alter the chemical atmosphere of the gut. Even within a single region of your body, multiple kingdoms of life intertwine. And none of it happens dramatically. There is no noise, no visible signal, just molecular exchanges unfolding in darkness. When antibiotics are introduced, the ecosystem shifts. Some microbes decline. Others expand to fill newly available niches. Most of the time, balance gradually re-emerges. Your microbiome is resilient, though not indestructible. Diet, stress, sleep, movement, all shape its composition slowly, like weather shaping a landscape. If you eat a wide variety of plant fibers, different microbial species flourish. Diversity tends to support stability. When diet becomes narrow or heavily processed, diversity can shrink. The landscape simplifies. But it can often be rebuilt, not overnight, not instantly, but gradually, through consistent patterns. Inside you are millions of microbial genes, far more than exist in your own human genome. These genes allow your microbial partners to perform biochemical tasks your own cells cannot manage. You are not genetically isolated. You are collaborative. Even at the cellular level, ancient symbiosis defines you. The mitochondria inside each of your cells, the structures that generate energy, evolved from bacteria billions of years ago. They are not invaders. They are remnants of an ancient partnership that became permanent. In that sense, cooperation is not something added to your biology. It is foundational. As you lie here now, perhaps thinking of yourself as a single body in a quiet room, there is something strangely comforting in realizing you are also a thriving ecosystem. Trillions of microscopic lives working steadily, not demanding your attention, not requiring conscious oversight. They digest what you cannot digest. They help educate your immune system. They shape metabolic signals. They occupy space so pathogens struggle to gain ground. They live, adapt, communicate, and shift, all beneath awareness. And you support them too. You provide warmth. You provide habitat. You provide food. It is mutual. Not perfectly. Not without tension. But functional, stable, and deeply ancient. The invisible life inside you is not something separate. It is woven into what you are. And as your breathing slows now, and your thoughts grow softer, that inner ecosystem continues its quiet labor. Molecules pass from cell to cell. Signals travel along nerves. Microbes ferment the remnants of your last meal. Immune cells rest in patient readiness. No rush. No drama. Just the steady unfolding of biology. You are not alone in your own body. You are an environment. Balanced. Alive. Supported by a hidden world that has always been there, working in silence. And tonight, it keeps working gently while you drift towards sleep. Right now, as your chest rises and falls in an easy rhythm, there are entire civilizations inside you adjusting to that rhythm. Temperature, oxygen levels, nutrient flow, everything shifts subtly with each breath, and the microscopic life within you responds. Your gut is not simply a tube for digestion. It is layered, folded, textured, lined with millions of tiny projections called villi and even smaller microvilli, expanding the surface area so dramatically that if stretched flat, it would cover the size of a tennis court. Along that vast internal landscape, microbes nestle into mucus layers secreted by goblet cells, hovering close to your intestinal lining without penetrating it. That mucus is not passive. It contains antimicrobial peptides, small defensive molecules released by specialized cells called Paneth cells. These peptides shape microbial populations, preventing overgrowth while allowing coexistence. The barrier is dynamic, constantly renewed, continuously negotiated. Some bacteria specialize in breaking down complex polysaccharides, long chains of sugars found in plant cell walls. Humans lack the enzymes to dismantle many of these fibers, but microbes carry entire gene clusters devoted to that task. Certain species within the genus Bacteroids, for example, possess what scientists call polysaccharide utilization loci, genetic toolkits that allow them to dismantle otherwise indigestible material. As they metabolize these fibers, they produce short-chain fatty acids that drift into your bloodstream and travel to distant tissues. Butyrate fuels colon cells. Acetate can be used by muscles. Propionate may influence glucose production in the liver. These molecules are not dramatic chemicals. They are subtle regulators, small signals shaping metabolic tone. Even cholesterol metabolism is influenced by microbial activity. Certain bacteria modify bile acids, transforming primary bile acids produced by your liver into secondary bile acids with distinct signaling roles. These altered molecules can bind receptors throughout the body, influencing lipid metabolism, energy balance, and inflammation. Your microbes also interact with tryptophan, an amino acid that feeds into serotonin pathways. They don't simply create mood, but they influence the availability of metabolic byproducts that interact with immune cells and gut neurons. The enteric nervous system, sometimes called the second brain, contains hundreds of millions of neurons embedded in the gut wall. It regulates motility, secretion, and sensation independently of conscious control. The microbes live beside this neural network. They release metabolites that can alter how those neurons fire. Signals travel along the vagus nerve toward the brainstem, integrating gut state with central nervous processing. Meanwhile, immune cells embedded in the intestinal lining sample microbial fragments continuously. Specialized dendritic cells extend tiny projections between epithelial cells to taste what is present. They present fragments to T cells, shaping immune tolerance. Regulatory T cells are encouraged to maintain calm in the presence of commensal microbes. This is not accidental. Your immune system evolved in constant contact with microbes. It expects them. Without exposure, immune development can skew. Overreaction becomes more likely. Allergies and autoimmune responses may emerge more easily when immune training lacks microbial diversity. Deep in the folds of your colon, microbial communities arrange themselves spatially. Some species cling closer to the epithelial lining. Others remain in the lumen. Oxygen gradients shape distribution. Near the intestinal wall, oxygen levels are slightly higher due to diffusion from blood vessels. Further inward, the environment becomes almost entirely anaerobic. And so anaerobic bacteria dominate, organisms that thrive in the absence of oxygen. They ferment. They reduce. They exchange electrons through metabolic pathways older than animals themselves. In fact, microbial metabolism preserves ancient biochemical strategies that date back billions of years, to early Earth environments where oxygen was scarce. In that sense, your colon is like a preserved chamber of deep evolutionary time. Beyond bacteria, archaeal species such as Methanobrevibacter smithii consume hydrogen gas produced during fermentation, preventing buildup that would otherwise slow digestion. By removing hydrogen, they make microbial fermentation more efficient. A cooperative exchange invisible to you but measurable in gas composition. And then there are bacteriophages, viruses that infect bacteria. They insert genetic material into bacterial cells, sometimes destroying them, sometimes integrating into their genomes. Phages can carry genes between bacterial species, including antibiotic resistance genes. They regulate population dynamics quietly, like unseen governors in a microbial parliament. Even the fungal residents. Species like Candida in low abundance interact with immune cells in ways that shape tolerance. When balance shifts, fungal overgrowth can occur, but in equilibrium, they are part of the broader mosaic. Your skin, too, has immune sentinels intertwined with microbes. Keratinocytes produce antimicrobial peptids. Sebaceous glands release lipids that certain bacteria metabolize into free fatty acids, influencing pH and inhibiting pathogens. Sweat contains salts and immune molecules that shape microbial composition. Every surface is curated by chemistry. In the mouth, saliva carries enzymes and immunoglobulins that regulate microbial communities. Dental biofilms are structured vertically, oxygen-tolerant species near the surface, anaerobic species deeper within. The plaque is layered, stratified, spatially organized. Inside your lungs, once thought sterile, low-density microbial communities exist, influenced by microaspiration from the upper airway. Even here, immune balance matters. Your microbiome also interacts with the endocrine system. Microbial metabolites can influence cortisol responses indirectly through immune modulation. Inflammation and stress signals loop through the body, affecting gut motility and permeability, which in turn reshape microbial communities. Stress changes the ecosystem. Sleep changes the ecosystem. Travel changes the ecosystem. Diet shifts it within days. And yet through all this flux, your microbiome shows remarkable resilience. After disturbance, diversity often gradually returns, not necessarily identical to before, but functional. Stability does not mean sameness. It means balance is restored. Your microbial genome, collectively called the metagenome, contains millions of genes. These genes encode enzymes capable of metabolizing compounds you would otherwise excrete unused. Some microbes even break down certain medications, altering their potency. Two individuals can respond differently to the same drug partly because their microbial ecosystems differ. You are not genetically alone in your own body. You are an assembly. Even the mitochondria inside each of your cells carry circular DNA reminiscent of their bacterial ancestry. Billions of years ago, an ancestral eukaryotic cell engulfed a bacterium that was not digested. Instead, a symbiotic relationship formed. That bacterium became the mitochondrion, the energy generator of your cells. Symbiosis built you. As you lie quietly now, microbes continue dividing approximately every few hours in your gut. Some species replicate quickly, others grow slowly. Entire populations shift in response to the remnants of your last meal. Fiber fragments drift through the colon. Microbes ferment them. Short-chain fatty acids are absorbed. Immune cells remain calm. Nerves relay quiet signals. There is no noise. Only molecular exchange. And perhaps the most peaceful truth of all is this: you do not have to manage it consciously. You do not command your microbes. You do not supervise fermentation. The ecosystem regulates itself through feedback loops refined by evolution. Competition prevents domination. Cooperation sustains function. Chemical gradients shape territory. Your body and its microbes have been partners for your entire life. And long before that, for the entire history of multicellular life. As your thoughts soften now, and your breathing becomes slower, the inner world remains steady. Tiny cells producing vitamins. Others modifying bile acids. Phage shaping bacterial numbers. Immune cells sampling and tolerating. An entire universe of life contained within your skin. Not loud. Not chaotic. Just working. Steady. Ancient. Alive. And you can rest, knowing that even in sleep, your invisible companions continue their quiet, essential work. Picture an Olympic stadium at night. Floodlights spill over polished lanes. The air hums with anticipation. A runner stands at the starting line, shoulders loose, breath controlled, years of training compressed into the space of a single moment. From the outside, what we see is muscle, lean, sculpted, efficient. We see lungs drawing in oxygen. We see heartbeats calibrated by repetition and discipline. What we do not see is the quiet ecosystem inside that body. Elite athletes do not only train their muscles. They cultivate internal environments shaped by diet, stress tolerance, recovery patterns, and movement itself. And inside them, trillions of microbes adjust to that life of intensity. Researchers studying Olympic-level athletes have found something subtle but consistent: increased microbial diversity. Diversity is not flashy. It doesn't make headlines. But in biology, diversity often means resilience. A system that can adapt to stress without collapsing. When scientists examined endurance athletes, they found elevated levels of certain microbial species rarely abundant in sedentary populations. One of the most intriguing examples is Veillonella atypica. After intense exercise, muscles produce lactate, a molecule associated with that familiar burning sensation during sprinting or climbing. Lactate travels through the bloodstream. And in some elite athletes, microbes like Veillonella appear to thrive on that lactate, converting it into short-chain fatty acids such as propionate. In experimental models, increased Veillonella abundance has been associated with improved endurance capacity. It is not that microbes run the race, but they may participate in the chemistry of recovery. In the gut of a marathoner, the environment is different from that of someone who rarely moves. Regular endurance training increases gut motility. Blood flow patterns shift during exercise. Hormonal pulses change the chemical landscape. Over time, these patterns shape which microbes flourish. Olympic cyclists, rowers, and long-distance runners often consume high-carbohydrate diets to replenish glycogen stores. These diets feed not only muscles but microbes capable of fermenting complex carbohydrates into beneficial metabolites. Meanwhile, strength athletes may consume higher protein diets, influencing microbial composition in distinct ways. Diet and training become twin sculptors of the microbiome. But it is not only endurance athletes who display microbial signatures. Elite weightlifters, gymnasts, swimmers each lives within a slightly different internal ecology, shaped by the metabolic demands of their sport. Anaerobic bursts, sustained oxygen uptake, recovery intervals, all alter internal chemistry. And microbes respond. Inflammation is one of the great silent variables in performance. Intense training produces microscopic muscle damage, part of adaptation. But elite athletes must walk a narrow line between sufficient stress and excessive inflammation. Some microbial metabolites, particularly butyrate, are known to interact with immune pathways that regulate inflammatory responses. A microbiome rich in butyrate-producing bacteria may support controlled inflammation and efficient repair. Again, this is not a single switch. It is a modulation, a gentle adjustment of biological tone. Stress hormones play their part too. Elite competition triggers adrenaline and cortisol. Acute spikes sharpen focus. Chronic elevation, however, can alter gut permeability and microbial composition. The highest performing athletes are not simply those with strong muscles, but those with systems capable of regulating stress and returning to equilibrium. Inside them, microbial communities mirror that balance. Even recovery sleep influences microbial rhythms. Deep sleep supports immune regulation and hormonal repair. Circadian alignment affects feeding windows, which in turn shape microbial activity cycles. Olympic training often demands strict routines, early wake times, scheduled meals, structured rest, all of which stabilize internal ecosystems. In studies comparing professional rugby players to non-athletes, researchers found not only greater microbial diversity but also associations between certain microbes and markers of protein metabolism. These athletes were consuming higher caloric and protein loads, feeding microbial populations adapted to that nutrient environment. The microbiome becomes, in a way, an echo of lifestyle. And perhaps most fascinating of all is that the relationship is bidirectional. It is not only that training shapes microbes. Microbes may shape adaptation. Certain bacterial metabolites influence mitochondrial efficiency, the tiny energy-producing structures within your cells. Mitochondria themselves are descendants of ancient bacteria. The partnership between human and microbe goes back billions of years. In an Olympic athlete, that ancient partnership is being pushed toward its limits. Oxygen delivery, lactic acid clearance, glycogen replenishment, immune modulation. All are optimized through training. But beneath those systems lies the quiet support of trillions of microorganisms processing fibers, synthesizing vitamins, modifying bile acids, shaping immune tolerance. They do not lift weights. They do not swim laps. They do not sprint. And yet, they participate. After a race ends and medals are awarded, the athlete rests. Muscles ache. Glycogen stores refill. Microbes ferment post-race meals. Short-chain fatty acids flow through the bloodstream. Immune cells clear cellular debris. Recovery is not dramatic. It is chemical. And in the darkness of the gut, the microbial world continues its work. Elite athletes often speak about marginal gains, tiny improvements accumulated over years. A fraction of a second. A slightly improved oxygen threshold. A more efficient stride. Inside them, marginal gains may also exist at the microscopic level, subtle shifts in microbial populations that support resilience, recovery, and metabolic efficiency. Not magic, not destiny, but collaboration. And perhaps there is something comforting in that image. The greatest physical performances on Earth, human beings pushing themselves beyond perceived limits, are supported not only by muscle and willpower, but by invisible ecosystems that evolved alongside us long before stadiums existed. As you lie here now, calm and still, your own microbiome hums quietly within you. It may not be trained for Olympic competition, but it is shaped by your habits, your meals, your sleep, your stress. It responds to how you live. And whether you run marathons or take slow evening walks, that inner world adapts to you, a steady, ancient, resilient. Trillions of microscopic lives working in silence. Supporting. Balancing. Participating. Even in greatness. Even in rest. And as your breathing slows once more, that ecosystem continues, unseen, uncelebrated, but essential. Quietly alive inside you. Before the stadium lights dim, before the breath slows and the body returns to stillness, there is another quiet frontier being explored, not on the track, not in the gym, but in laboratories where scientists peer into genetic code that does not belong entirely to us. In recent years, researchers have begun to wonder whether the invisible ecosystems inside elite athletes are not only shaped by training, but whether they might, in subtle ways, help predict performance potential. This idea must be approached carefully. The microbiome does not determine destiny. It does not decide who becomes a champion. Human performance is a layered tapestry of genetics, environment, discipline, opportunity, psychology, and resilience. And yet, patterns have emerged. When scientists sequence the gut microbiome of endurance athletes, they sometimes find elevated levels of bacteria capable of metabolizing lactate or producing metabolites associated with metabolic efficiency. In power athletes, microbial signatures sometimes reflect dietary adaptations that support protein metabolism and muscle repair. These patterns are not guarantees. They are tendencies. Researchers now analyze stool samples using high-throughput sequencing techniques, identifying bacterial DNA fragments and mapping microbial communities in extraordinary detail. Each individual's microbiome becomes a kind of ecological fingerprint, dynamic, personal, shifting over time. Some scientists speculate that certain microbial compositions may support enhanced recovery, reduced inflammation, or improved fuel utilization. Could the presence of specific butyrate-producing bacteria correlate with faster muscle repair? Could higher microbial diversity signal greater resilience under physiological stress? These are questions still unfolding. In controlled studies, transplanting microbiota from high-performing animals into sedentary ones has, in some cases, altered metabolic outcomes. In humans, however, the picture is far more complex. Microbiomes adapt to training, but training also adapts to physiology. It is difficult to separate cause from effect. Does the microbiome help create the athlete? Or does the athlete's lifestyle cultivate the microbiome? The answer may be both. Some research groups are exploring whether early microbial patterns in youth could correlate with metabolic flexibility, the ability to switch efficiently between carbohydrate and fat metabolism. Others are studying how microbiome diversity relates to oxygen utilization and VO max, a measure of aerobic capacity. There is even discussion, careful, cautious discussion, about whether future sports science might include personalized microbiome modulation. Not doping. Not shortcuts. But tailored nutrition plans designed to support beneficial microbial communities already present within an athlete's body. Yet even here, humility remains essential. Microbiomes are not static codes waiting to be decoded. They are living systems that change with diet, sleep, stress, antibiotics, travel, and environment. A test performed today reflects the ecosystem of today, not necessarily tomorrow. And perhaps that is the most grounded truth. The microbiome does not predict greatness in isolation. It participates in it. It reflects it. It adapts alongside it. An Olympic athlete's microbiome is not a magic formula. It is the ecological imprint of years of structured training, disciplined nutrition, controlled stress, and consistent recovery. The ecosystem inside them is not separate from their effort. It is shaped by it. And that means something quietly hopeful. Because ecosystems can change. They can diversify. they can strengthen. As we move towards stillness now, imagine the idea of potential not as something fixed in your genes alone, but as something influenced by relationships between your cells and your microbes, between your habits and your chemistry, between your movement and your metabolism. Inside every human body, there is adaptability. And inside that adaptability, there is possibility. And so, as the stadium lights dim in your imagination and the roar of the crowd fades into something softer, what remains is not the medal, not the finish line, not the flash of cameras. What remains is breath. A body returning to stillness. And inside that body, an entire hidden world settling into its own quiet rhythm. It is easy to think of human achievement as something singular. One person, one will, one set of muscles straining toward excellence. But the deeper truth is quieter and far more beautiful. No human body is alone in its effort. Beneath every stride, every lift, every dive into cold blue water, there are trillions of microscopic lives adjusting, responding, participating in ways so small they escape our senses. Long before humans learned to run for sport, before we carved stadiums into stone and timed our cells with precision, microbes were already here. They shaped the chemistry of early Earth. They filled the oceans. They altered the atmosphere. They learned to cooperate, to exchange molecules, to share genes. And eventually, they entered into partnership with larger life. That partnership became us. Inside your cells, mitochondria, once free-living bacteria, generate the energy that allows muscle fibers to contract. In your gut, microbial communities transform food into signals and fuel. On your skin, invisible guardians maintain quiet order. You are not separate from these histories. You are their continuation. Even now, as you rest, the chemistry of movement lingers in your tissues. Glycogen replenishes. Immune cells repair microscopic tears. Hormones return to baseline. And within the folded darkness of your intestines, microbes ferment the remnants of a meal, producing gentle molecules that flow outward like soft currents through your bloodstream. There is no applause for them. No anthem. Just steady work. If you imagine zooming outward, beyond the stadium, beyond the city, beyond the planet itself, you would see Earth glowing softly in the vastness of space. And on that small blue sphere, every human who has ever moved, loved, competed, struggled, rested, each one has carried this same invisible ecosystem within them. A collaboration older than language, older than civilization, older than the idea of self. When an Olympic runner crosses the finish line, what you are witnessing is not just discipline and muscle memory. You are witnessing billions of years of evolutionary partnership compressed into a single human moment. And as your breathing slows now, you can let that partnership hold you. The work continues quietly. The chemistry continues gently. You are never alone inside yourself. And you can rest.